Well, good afternoon, good morning, good evening to everyone listening in from around the world, and welcome to our European MedDRA user group webinar. First of all, just a few housekeeping points here. We will be answering questions on the presentation later on in this session. So feel free to drop your questions into your dashboard with the instructions on the screen there. You can pop out your control panel located in your upper right of the screen, type your questions in the question box, and then submit by sending send. We'll try to get through as many questions as we possibly can, but obviously there may be a large volume. Feel free to ask those questions throughout the webinar, and we'll get back to you at the end. Okay, one of the FAQs that we always get is, will I be able to get a copy of these slides? Well, you can actually see the slides in your handout section in the panel to your right, and you can pick them up already from there. They'll also be available on the MedDRA website. And also the recording of this session will be on the YouTube channel, and also again there'll be links on the website later. So this lovely set of people are my colleagues in the European Industry MedDRA User Group Steering Committee, and we have been putting together these webinars and presentations over a number of years. Today we have myself presenting and as a facilitator, and we have Anne and Christina who will be presenting the session today. Okay, we're going to ask a few polling questions initially, just to get some facts about who's actually on the line. So this is our first question for you. From which region are you attending this webinar? A, Africa, B, Asia or Oceania, C, Europe, D, Latin America and the Caribbean, or E, North America. And you'll just have a few seconds to answer that poll question, and then we'll share the results with you.
Okay, it looks like votes are starting to stop. I will close the poll and share the results.
Okay, lots of Europeans on the line. Great. Thank you for that. The next poll question. What type of organization do you work for? Whoops. Pharmaceutical development, manufacturer or distributor organization, medical device development organization, a CRO, regulatory authority, or other.
All right, I will close this one as well and share our results.
Okay, great. Oh, quite a few others in there. Be interesting to know what the others are. Thank you very much, and we'll move on to the next poll question. What is your role, position? Is it drug safety, pharmacovigilance, clinical research or operations, coding, stats programming, informatics or data management, or other?
And here are our results.
Not surprisingly, because of the topic today, many of you are in drug safety and pharmacovigilance. Very good. Right, and then I think this is our final poll question, which is, do you have any experience with signal detection? And it's yes or no.
All right, it looks like our votes are trickling off, so I will close and share the result.
Ah, interesting. 2/3 with a yes and 1/3 with no, and we'll be hoping to learn a few things from this presentation. So I hope we do well with that. Okay, so first, right now, thank you very much for that. We'll now move on to the agenda. So it's a very short agenda today. We just have the one topic where we will be receiving a presentation on MedDRA aspects in safety signal detection and evaluation. And then we'll have the Q&A and we will then move on to talk about the next face-to-face EU user group meeting. So without further ado, I'm going to introduce our presenters today, who are going to be talking about MedDRA aspects in safety signal detection and evaluation. We have Anne, who is a former committee member and is now with the MSSO, and we have Christina from GSK. I'll hand over to you, Anne.
Thank you, Ian. Good morning, good afternoon, and welcome everybody. My name is Anne Jüllenswert, and as Ian just said, I recently joined the MSSO in the position of an international medical officer, meaning that my main responsibility is with the terminology maintenance nowadays and as well as SMQ maintenance. But before that, I worked 13 years in a pharmaceutical company, a medium-sized company, Grünenthal, with headquarters in Germany. And I worked in pharmacovigilance, all from first-in-human studies until products in the post-marketing. And in my role at Grünenthal, I was also responsible for the conventions and processes associated with the MedDRA use in Grünenthal. That is my background. Thanks a lot, and over to Christina.
Hello, everybody. I'm afraid, I hope you can hear me, but you can't see me because I've had IT problems and I've had to use my home desktop, which doesn't have a webcam, so I can hide behind the anonymous screen. I've been in GSK for about 30 years and been mainly in pharmacovigilance, initially with clinical trial products as well as with post-marketing, but in more recent years, mainly post-marketing. I've been associated with MedDRA for, I've lost track of time, since it was adopted by ICH. And within GSK, I've also been helping with the MedDRA processes, implementation of SMQs, etc. when they first came out. I was also in the SMQ working group for several years. Within the ICH structure, I've been on the MedDRA Management Committee for about a decade, 2 decades, sorry, and on the Points to Consider working group. Anne also didn't say that she was on the Points to Consider working group for a while. At least if she did, I missed that. So anyway, without more ado, let's go into the presentation. Over to you, Anne.
Okay, thank you. So let's start. As of first, we have a disclaimer that this information is our own. It's not the view of GSK, the MSSO, or Grünenthal for that reason, and you can read it at your convenience later on. So we were thinking that we might face quite a heterogeneous group of people with different knowledge of signal detection. And I was surprised to see that there are so many people working in pharmacovigilance joining today, but still good to know that 70% say they have knowledge of signal detection and 30% don't, because we structured our talk a little bit to accommodate everybody and wanted to start with what is a signal and what does it mean. And so for those who are very familiar with this definition and signal detection overall, please bear with us. But for those who are not, I hope that we can give you some background information. It will be very short and not a deep dive into signal detection, but just for you to be able to follow us later on. So a signal is any information arising from one or multiple sources, including observations and experiments, which suggests a new potentially causal association or a new aspect for a known association between an intervention and an event or a set of related events, either adverse or beneficial, that is judged to be of sufficient likelihood to justify verificatory action. So what does this mean? At first, it means the signal does not mean that there is a new risk for the product. It's just a first hint that we need to do something to see whether we have a new risk. It also means a signal can be detected in one or in several sources. And we come to potential sources in the next slide. It also says that a signal can be adverse or beneficial. So it's not only the adverse reactions which we can find. There are also some examples where something beneficial was found first as a signal. And then very importantly, the detected information may suggest a new potentially. So it's not a causal association, but there is the potential that there is a new causal association. And then also very important, the information is sufficient to justify verificatory action. So we see a lot of things, but of minor relevance or with so little information that it's not yet justified to put a lot of effort into a signal evaluation as a next step. We have tools available with the aim to help us in decision making, but in the end, a signal assessment always needs medico-scientific assessment. And that is important to decide whether verificatory action is justified or not. So what are potential sources of signals? We see signal detection as a multidisciplinary approach, and we consider data sources as, of course, spontaneous individual case safety reports and the pharmacovigilance databases, the company database, but also the national and international pharmacovigilance databases, which are Eutrovigilance, for example, the FDA FAIRS database, or the World Health Organization's Vigibase. But we see also other data sources like non-clinical data, for example. Even if the drug is older, new non-clinical data can also lead to signals. Of course, clinical trial data. And that can be clinical trial data during the clinical development, but it could even be clinical trial data of a similar drug, which points into a new direction, that there might be something which could also be a class effect. So clinical trial data is also very broad. We have potentially registries or epidemiological and claims data or other data sources. The scientific literature is a wealth of information which can lead to signals. We also used to look at the information published by regulatory agencies on their web pages, because they also start looking into signals, and that is an early hint to also start evaluating whether something might be behind that. And then we can always look at comparator or class labels and see whether other drugs, other companies have detected signals for their drugs which are closely related to the product which we are responsible for. But it's also important to note that not all data sets are available or relevant for all products. So for very old products, you might not have the same non-clinical data package which you expect for a new product. Or if you're really early in development, you will not have the spontaneous individual case safety reports, for example. So it's really dependent on the product, what kind of data sets you can expect. And then, of course, not all data sources are accessible for all experts, meaning that if you have a generic drug, there is a lot of a wealth of data available for that drug, but you might not be the one holding that data, for example, the complete non-clinical package or the clinical trial data. So even for drugs where there is a lot of data, it's always the question whether you can access the data or not. So let's also discuss challenges in signal detection. I like this picture quite a lot because it shows nicely the limitations we have in pharmacovigilance or in clinical development in general. So what you can see here is the time during the clinical development when people are included in randomized clinical trials, and you see there's a small number of patients until you reach the time when you apply for marketing authorization. You get your license, and then the clinical trials are usually continued to look at other populations which were not included in the first wave of patients. You might start doing observational studies and registries, and all these experiments have quite a good data quality. But then you also start gathering the post-marketing data, and you see here that the axes are divided because your post-marketing data will be a lot more than what you ever can collect in clinical trials. But the data quality is different. So you don't know how many patients have been treated. You don't know what other drugs they had. You often don't know what kind of other medical history they had. So you're treating a very broad population, but the quality you collect is not as high as in the clinical trials, where you treat a smaller, very well-defined population in a very well-defined setting. And then this is about the patient populations and the data quality which you collect over time. And then at certain times, we have different stages of knowledge. And there are things that we know that we know. So known knowns is things where we know, okay, we have this ADR X, and it occurs in 10% of the patients. That is something which we know, which we can put risk minimization on. Then there are known unknowns. That is usually saying that we have, for example, a population, pregnant women or children are often those populations where the clinical which are excluded from clinical trials. So we know that we have limited knowledge in those populations. And that is the known unknowns. We will need to collect more data in post-marketing to compare that data to the knowledge which we had to see whether there is something new coming up out of those known unknowns. And then we have the unknown unknowns, and that is everything where we don't have a clue that it might happen and we're not prepared for it. And signal detection is on all these things. So we usually have a signal detection process where we plan what kind of data we look at. And some part of it will be, we know that we have a gap in knowledge on a certain ADR. So we would like to get more knowledge on that ADR, and we define a search strategy for looking at a certain medical topic, or we define a search strategy for looking at a certain population, and we define in which frequency we would like to look at that data. But then on the other hand, we also know that we have these unknown unknowns. So we also plan to look at all the data to see whether we find something in that big haystack of data, which we then call a signal. So we have a planning phase where we set up our signal detection routine. We define what data to look at, which frequency to look at the data. Then we get the data, we review the data, we decide whether we see a new signal. If we don't, we document, and then we repeat the complete process. In the case we decide that there is a signal, we go into the signal evaluation process. So we validate the signal. We say, yes, there is sufficient information to go a step further to do this verificatory action, which we call signal evaluation. But during the validation or as a next step, we often do prioritization. So defining how important is that topic and how fast do we need to do the evaluation. And then we evaluate. After this evaluation, we take a decision whether we see a new risk, whether we need to take further action, and then we manage the new risk. Or, of course, the outcome can also be that there is not enough information yet, so that we don't consider this a new risk. Our process is also open to include signals from other sources. So we cannot plan for a literature to be published and we find it. Of course, the literature review can go into this cycle, but we can get signals from other sources. And these, once we have identified a signal, we will follow the same steps. We will validate it. Often companies have a validation body, which is a bigger group of people saying, yes, we all see that there is enough evidence to do the next step. Then we do the prioritization. We go into the evaluation, take a decision, and manage the signal. So we discussed signal sources. Then we have several signal detection methods, and we distribute these into qualitative and quantitative. And qualitative, and both are important, but they have different use cases. So qualitative signal detection usually means you're looking at individual case reviews, sometimes also on an aggregate level, like line listings or summary tabulations, and you're looking at literature. The quantitative, on the other hand, is you're looking more on statistics like reporting rate, trend analyses, or a little bit more advanced statistics like data mining, which can be done, for example, on the Vigibase or on the Vigilance database. But big companies often calculate these statistics also on their databases. Grünenthal's database was quite small and very heavily focused on a certain product portfolio. So we did not do the data mining on our own safety database, but we used these two big databases for quantitative signal detection methods. The quantitative is more for signal generation. So you're looking at this, finding the needle in the haystack. You're calculating a certain ratio to determine that you're just passing a threshold, and then you need to look in more detail. And Christina will show you some of these things in her part of the talk. The qualitative signal detection methods are very helpful if you have a topic which is rare in the background population, where just a few cases already can lead to a signal or to the evaluation of a signal. So keeping this in mind will also help when we discuss later on. And then I just wanted to highlight, because we are talking about MedDRA, that there are a lot of supporting resources from the MSSO, which can be found on the MedDRA.org website. And the first thing I wanted to highlight is that there is a points to consider document, which is an ICH approved document on data retrieval and presentation. And you can find it if you open the MedDRA.org website. There's the support documentation. And then you have the MedDRA data retrieval and presentation points to consider document. This document is the maybe better known points to consider for MedDRA term selection, is released with every yearly MedDRA update with the complex versions, the .0 version. Then the MSSO is also holding webinars on a recurring basis on data analysis and query building with MedDRA, as well as one webinar which is called Standardized MedDRA Queries. There are sometimes also face-to-face classes for these two topics, And what I find really helpful is the YouTube playlist, where you can also find these webinars, and you can even choose which presenter you like, whom you want to listen to, and you can watch it at your leisure. Also to highlight is that there are mappings to other terminologies, and there's quite a lot of mappings already. There's to CTCAE, ICD-10, SNOMED, IDMRF, and an ICD-11 mapping is underway. So in case you are evaluating a signal for something where you need to map data, this might be a helpful resource as well. And with that, I hand over to you, Christina.
Thank you, Anne. I'd like to show a few examples of how signal detection is done in GSK, and it will give examples of where we have a tool that enables us to look at the predefined thresholds. Can I have the next slide, please, Anne? Thank you. So first of all, how is a signal generated within GSK? For start, we have a list of MedDRA terms of interest, and these are groupings of MedDRA PTs, or even if you have a suitable HLT, you can use that as well for grouping terms. And this is for incoming case awareness for any post-marketing reports or even clinical trial reports. If you have got a huge trial and you've got a lot of SAEs coming in, you can use your terms of interest. And we have two sets of terms of interest. One is common to all products, which is basically drug-related events that have got public health impact. You know, and these are the ones that will mean re-evaluation of benefit risk, potential healthcare professional letters, or dear investigator letters, might lead to urgent safety restriction, or even at the worst withdrawal from market. So these with public health impact are the ones that we need to be aware of quickly, in common for all products, and they require talking to regulators as well if you do find one of those signals. In addition to that, we have product-specific terms of interest for regular ongoing monitoring, and these product-specific ones may be related to your patient population, or it may be related to the mechanism of action for a particular product. And it might also be class effects that you might be wanting to look at for similar products. So these two sets of terms of interest are built into our signal detection tool. If you wish to have more information about terms of interest, one of the previous webinars that this group produced in January of last year, there was a versioning webinar, and you will find a reference to terms of interest if you go to that recorded webinar on the MSSO website. This is the January 2022 user group webinar. We also look at ongoing literature review in GSK for that product, and obviously the special focus for literature review will be using the terms of interest, and that's a special focus, but it's not exclusively on the terms of interest, because there might be something coming out of literature that you'd never thought of before. This is the unknown unknowns that Anne was referring to, that just come out of the blue and catch you unawares. And sometimes they are because somebody has a very similar product that you had not been aware of, and they found something and they publish. So that can sometimes come out as an unknown unknown. For the signal detection tool, we usually look at periodic aggregated data, whereas the first bullet point on this slide, where we're using terms of interest for incoming case awareness, those are individual cases. And individual cases are looked at on a daily basis if you wish, because it pops up on the tool as and when the case enters the safety database. But to look at periodic aggregated data, we would use a separate signal detection tool to the one that looks at incoming cases. The signal detection will look at periodic aggregated data, and that you can choose to do it over a month or fortnightly or weekly, depending on how active your particular product is. Those of us in pharmacovigilance know that shortly after the launch of a product, you usually get a lot of spontaneous reports, post-marketing reports. And after 2 years, that level of reporting tends to generally fall off. So if you've got a very old product, perhaps once a month is enough. If you have something shortly after launch, you might want to look at it more frequently than once a month. And for extremely old products that perhaps sales are not brilliant and you've got very few reports, you might want to do it every 2 or 3 months. It really just depends on your particular product. You can also look at just the background rate. In other words, what is coming in for that month versus the background rate, or look at spikes and aberrations. For spikes and aberrations, what I mean is for a particular product, you suddenly get a spike, meaning, you know, an abnormal number of reports of that particular preferred term in a month. So say if you launch new packaging, child-resistant packaging, which is more difficult to open, the elderly or those with disability will find it more difficult to open the package, the new package. So for a while you may have product complaints coming in, and that would be a spike. Or they're saying, oh, I've missed my dose because I wasn't able to get into the package to take my dose. So those are the sort of spikes that you could get, and they come up and they will persist for a few months, and it might go away as the patients become more accustomed to the packaging, or as education materials are rolled out to show them how to open the package to get their medication. And within GSK, because we have a very large safety database, initially it was well over 300 products. The company has now divided into two, consumer health and pharmaceuticals, apart from vaccines. So the numbers in the actual database of products has shrunk a bit, but it was initially over 500. No, you still have well over 300. So it's a pretty good way of comparing within the products. And because they are not one therapeutic area, they're a myriad of therapeutic areas, we can look at that particular database for cumulative figures. So you're comparing your particular product against all other products in the database. Can I have the next slide, please, Anne? Thank you. We've got a very, very high volume of post-marketing case reports. You're talking tens of thousands coming in. And for one particular product, we've got over 100,000 reports, and that was 10 years ago. Goodness knows what the figure is now. So we are able to compare case products, but we also check the internal signals against eutrovigilance and against FAERS in the signal detection tool. We were looking at two different tools. Our older tool looked at MedDRA PTs as well as SMQs for the signal. But we found that the signaling within an SMQ, when we investigated it, was usually due to a single PT within that SMQ. And when we looked at the signal score, we found that for whatever reason, it was diluted when using the SMQ, because one PT was signaling high and other PTs were signaling low. So by the time you use the whole SMQ, the signal score was slightly lower using the SMQ compared to the single PT that was signaling. So the current tool now only uses the PTs rather than the SMQ for signaling, but the reviewer has got MedDRA awareness, and if you see a signal in a PT, you look at the other relevant terms and look at the SMQ as well before you make further decision. Can I have the next slide, please? Thank you. This is a pictorial demonstration of what it looks like. The neutropenia is the term we're interested in, and obviously agranulocytosis is one particular PT, so it's within the HLT of neutropenias. It shows us blue squares on the left-hand side where you've got blobs signaling very near zero. Within this particular database, signaling database, EB05 of 2 is where there is a signal score, so that's the threshold. So anything that gets to 2 or above will show up as red. Anything that's blue is not signaling. So we've got agranulocytosis, neutropenia, afebrile neutropenia, and granulocytopenia. So they're fairly similar terms within the HLT of neutropenias. The data source for this one is the internal database, OASIS. This is a period of a month, and this screenshot was taken before the change to MedDRA 26.1. So at the time, we were using 26.0. Can I have the next slide, please? This is a trend graph for the same PT, agranulocytosis, and it shows you the change over time from May 2018 right through to May 2023. And that's within the same database. And the signal is flat throughout. The threshold, as I said earlier, was up at 2. So this is running just above 0. So basically, it's okay in this particular database. We'll move on to the next slide, please. This is where we check it against the same PT and the same similar PTs, but using Vigilance, which is the European EMA's database. Same time period and the same MedDRA version. You can see that the blues are not all running along a straight vertical line. There's some scattered over to the right-hand side, but it's still not signaling. Now, within the Vigilance database, the scoring system is different. Instead of using EB05, they're using ROR, and that is a reporting ratio, and their threshold is 1. So if it's over 1, it will show up as red. Can I have the next slide, please? This shows a trend over time, and within the Vigilance database, we can't see as far back as 2018. So here we've only got 2020 through to April 2023. There's the numbers of cases bumps up and then bumps down again. Well, I haven't got the details, but those in pharmacovigilance will realize that you can sometimes have a duplicate, and we're only talking 1 or 2 cases here difference, because this is the case number N. And it might just be that the report came in and then they found duplicates, so the numbers went down. And it does sometimes affect the reporting ratio, so you can bump up and down, but it's fairly stable and it's still under 1. So that confirms that the signal in the in-house database is not a safety signal. So the next slide will show what the FDA is doing. Fairly similar cluster. The FDA uses EB05, which has a threshold of 2, similar to the GSK internal database. Here we're looking at the same month, and the data is a little bit older in FAIRS, because you see it's from last year to January. So we're looking at a quarter, slightly different time period, and also it's MedDRA 25.1 instead of MedDRA 26.0. But nonetheless, it's still not signaling. And if I could have the next slide, please. Thank you. It's fairly flat as well. So for agranulocytosis and that particular product, we don't have a concern at the moment. Can I move on to the next one? Thank you. Now this is an example of where the PT has exceeded the signal score threshold. And the next slide shows the GSK database for the PT tinnitus. And we're looking at similar terms within the HLT, inner ear signs and symptoms. So you've got tinnitus, you've got motion sickness, vertigo, and vertigo positional. 3 of these PTs are sitting under the threshold of 2, but tinnitus is way over and signaling that there is something going on there. And this is within the GSK database tool. Tinnitus, by the way, is a labeled event, an expected event for this particular product. So it's not really surprising that it's signaling well above the threshold. Can I have the next slide, please? Thank you. You can see that this has been signaling from May 2018 right through to the current, the last point at which this screenshot was taken. And it's been consistently high over the threshold of 2. The next slide will show the eudravigilance database. Tinnitus is also signaling there using their scoring of 1 being the threshold. And this is also a similar period in time to the GSK database. I remind you, this is a listed event. It was discovered during early clinical trials and it's been in the product label ever since. And this is an old product of over 20 years on the market. The next slide will show the trend over time with eudravigilance. Now you might wonder why it's actually falling despite a fairly steady but slow rise and then leveling off. But the score is falling. But the score is based not just on sheer numbers, but it's also based on the background rate of tinnitus being reported for other products within the eudravigilance database. So it might just be that other products within that database are beginning to report tinnitus. So the signal score for this particular product compared to the rest of the database could fall, although the actual numbers of cases for this product for tinnitus is fairly steady. Can I have the next slide, please? We're going to look at it over FAIRS now. It's signaling there as well. But as I said before, we knew that this was a labeled event, and so that is not surprising. Let's look at the trend in the next slide for FAIRS. It's stable, but it's very, very high signaling. This is the threshold of 2, and the numbers of reports are fairly stable, which means that there is really no increase in reporting because these are cumulative numbers. The next slide, please, Anne. Thank you. So after a signal is identified, what do we do? First of all, you need to ask yourself, what is the question you're trying to answer? So for something like a PT of long QT syndrome, which sits in a cardiac disorders SOC, you've got to consider that there are very similar PTs sitting in the investigation SOC. So electrocardiogram QT prolonged will not actually show up in the graphs that I was looking at earlier, because those graphs looked at the PT and the HLT around that particular PT. So a graph for long QT syndrome will only look at the relevant HLT within cardiac disorder stock. It will not show the PT electrocardiogram QT prolonged that's sitting in the investigation stock. So whoever's doing the signal detection has to think MedDRA and go and look at the investigation stock. In addition to that, you then have to have the medical thinking of, do you really want to look at other arrhythmias, other conduction defects, or repolarization disorders for that particular product? Are they in scope, or are you only interested in long QT? The other medical consequence of long QT is torsade de poids, which is a really nasty arrhythmia that can lead to sudden death. So do you want to look at torsade as a PT as well in your signal detection? And do you want to look at sudden death terms in your signal detection? So those are the thoughts that have to go in, knowing the medical condition, but also knowing MedDRA and looking elsewhere, not just sticking to your plots that come out of the signal detection tool. Another example is a published case described interstitial pneumonitis. That sits as an LLT in MedDRA, and the PT for interstitial pneumonitis is interstitial lung disease. There is also a similar PT for pneumonitis itself, and there's also an SMQ for interstitial lung disease. So should you search the SMQ instead? Because the SMQ includes both the PTs of interstitial lung disease and pneumonitis. But the SMQ also includes other things like radiation pneumonitis and other sort of causality type terms. So if your product is not some kind of isotope used in scanning, would you really want the PT for radiation? There are also exposure to different types of dust, industrial things that can cause pneumonitis. Would you want those PTs? So is the SMQ too broad? So you really need to consider MedDRA, consider what you're looking for, maybe use the entire SMQ, but maybe use a modified SMQ, which will, for instance, within GSK, we call it a GSK-MQ, or just modify it for that particular search. So you have got a lot of decisions to make once you identify a signal. And I think I'm near the end of my presentation. Can you have the next slide? Oh, yes, one more. So basically, you need to review all your available data. Literature search may not be MedDRA coded. The publishers and authors are not necessarily using MedDRA. They are physicians, and they could be using any medical term that comes to mind or whatever is in common medical use. So you need to look at the keywords and think, don't just search MedDRA terms because they may not be MedDRA coded. Epidemiology, it's often in ICD or SNOMED, depending on the data source. And as Anne's already said, there are mappings on the MSSO website for several other terminologies, and certainly ICD-10 and SNOMED are there. Clinical trial data, recent clinical trial data will be MedDRA coded, but if you go back 30 years, like some of my older products, the clinical trials were done in the 1970s and 1980s, long before MedDRA was used for clinical trials. So is your very old data, has it been migrated, or has it been left sitting in the old terminology? You need to consider that, especially if you're trying to merge newer data versus the older data, one MedDRA coded and one not MedDRA coded. And we compare against ultravigilant cases, and we look at the narratives in ultravigilant cases as well. And at the moment, although we can see the signal scores for FDA, we can't easily get the narrative. So we can't look at the narratives there. So I think that really is the end of my pack, and back to you, Anne. Thank you very much.
Thank you, Christina, and thanks for the nice handover, because what I'm now showing, what Christina showed you, is more the planned signal detection, which is repeated on and on again. And with the next part, we wanted to show you what are you going to do if you find a signal, and then how do you use MedDRA in signal evaluation. So what I'm going to show you two examples, and one is for the non-steroidal anti-inflammatory drugs and a topic called drug reaction with eosinophilia and systemic symptoms, DRESS. This is a signal which came from a competent authority. And the first thing which you need to do is usually you need to understand the medical topic. And especially for drug reaction with eosinophilia and systemic symptoms, this is something very rare. And even if you're a doctor, you might never have heard about it or never seen a patient with that symptom. Then the other thing you need to consider is what kind of data sources do I have available, which will be helpful at looking at this topic. And then it's very important to set up a search strategy. So let's start with trying to understand the medical topic. And DRESS is a severe adverse drug reaction. It has very extensive skin rash, but in addition to that, it has visceral organ involvement, lymphadenopathy, eosinophilia, as already announced in the name of this syndrome, and atypical lymphocytosis. It is known with different names. One of the others is drug-induced hypersensitivity syndrome. And the clinical presentation, although it's one of the serious skin reactions, it can be very heterogeneous. The disease course is often prolonged, and even despite cessation of the offending drug, it can flare up again. In pharmacovigilance, we're often looking at latency, meaning when did the patient start taking the drug and when did the reaction occur? And for this reaction, it's prolonged. So the first symptoms usually occur between 2 to 8 weeks after starting the new drug. What is also reported is a reactivation of latent human herpes virus infection. And it is approximately 1 to 2 per 100,000 patients per year, so a very rare event. And having said this, we have good chances of certain data sources. So we know it's drug-induced. We know it has a severe extensive skin rash. That means doctors see it, the patient directly realizes something is wrong. And because it's so impressive, it's often reported. It's a rare disease, so we are likely to find it in PV databases, and that these will be a relevant source. The other relevant source will be literature, because these cases are often published. And for the clinical trials, although they have a very good quality of data, most likely they will be too small for relevant assessment. So we identified our data sources, PV databases and literature as the main sources. And then the next step will be to decide how to retrieve the data. And what I often do is that I just open the MedDRA browser, put in the name of the reaction, and you see there is a PT drug reaction with eosinophilia and systemic symptoms. So I already have a hit. If you click on the term, and I already did this for this screenshot, on the right to the term, another window opens, and it gives you more information on that preferred term. So you see the MedDRA code, you see the SOC names of it has a primary SOC and a secondary link. But then what is really important and interesting is it also gives you an overview into which SMQs this PT is included. And you see here the PT is included in the SMQ drug reaction with eosinophilia and systemic symptom syndrome. It is also included in the SMQ eosinophilic pneumonia, in the SMQ hypersensitivity, and then into SMQ severe cutaneous adverse reactions. So now we have a good choice. And I think that there are two SMQs which sound quite promising, the DRESS SMQ and the severe cutaneous reactions SMQ. And my next step will then be to go to the SMQ introductory guide to see the in- and exclusion criteria for these SMQs, to be able to judge whether the SMQ is designed for my question or whether it is not appropriate at all for my question. The SMQ introductory guide can also be found on the MedDRA.org website. It is published with every MedDRA update and available for every MedDRA version. So this is also the introductory guide for version 26, because we designed these slides already in the spring of this year, when version 26.0 was still the valid version. And the SMQ introductory guide is built up that it gives a general overview of what SMQs are, what they can be used for, but then it goes into all individual SMQs and provides description, which usually starts with a definition, and then you have the in- and exclusion criteria and some certain further information on how the SMQ is thought to be implemented or to be used. So if you are going to the section on DRESS, it's 6 pages long. That's why I just put you the first page, but it gives you a definition of the syndrome. It also tells you a little bit about drugs which have been associated with DRESS, and it continues with in- and exclusion criteria. And then here you can still see that this is the rest of the inclusion criteria. Here you have the exclusion criteria. But I put you this slide because what is very interesting is that there is an algorithm related to this SMQ. And an algorithm means that there are certain search terms which are relevant independent of any other term, which are the category A, the narrow search terms. It's just 3 terms, and I'm going to show you those to you. But then there are a lot of other terms which are related to organ damage, which are related to fever, which are related to lymphadenopathy, or to the hematologic abnormalities. And just a combination of certain of these terms will lead the case, if it is not anyways identified through a narrow search term, to be added to your search. So you can see here on the side that there are the narrow search terms, or that you can have a combination of the category B with 2 or 3 from C, D, or E. So that is how you would implement this. It's not the usually way you use an SMQ, and some people might not be very familiar. So my proposal would be that before you just request such a search to be implemented, discuss it really with your database retrieval personnel to see that they know how to correctly implement such a search. It might be that they can do it. It might be that they cannot do it for some reasons. At Grünenthal, we were able to do it. I always discussed it because it's just not what people are very familiar with. Then we also said that there was another SMQ, severe cutaneous adverse reactions, which includes stress, which sounded interesting. So we see here in the definition that there are more than one severe cutaneous adverse reaction included. So we have erythema multiforme, Stevens-Johnson syndrome, toxic epidermal necrolysis, then the DRESS, as well as acute generalized exanthematous pustulosis. So the question here is really, again, what do I need to evaluate? If your drug is already labeled for Stevens-Johnson syndrome, you've done several analyses of toxic epidermal necrolysis, you've reviewed erythema multiforme, you might want to just focus on DRESS. But if this is the first hint that your drug might be involved in severe cutaneous adverse reactions, you might also decide to go broader and to look for all the severe cutaneous adverse reactions. So this is really a medico-scientific decision which you need to take when you're looking at this, at your topic. So then the next question would always be, what is included in the SMQ? And you have several ways of seeing that. So one easy way is to go into the MedDRA browser again, and usually you're looking at the SOC view. But you can change that browser view to SMQ view. And if you do this, you have the you also have a search function, which I did not use this time, but you can open the SMQ, like you can open the hierarchy, and it shows you all the terms, not on this screen because there were too many, but it shows you the terms which are included in the SMQ. And you see here the blue PTs are the narrow terms, and the red ones are the broad search terms. Another good way to look at SMQs is the SMQ spreadsheet, and I really like this resource. You might not be familiar with it. It's contained in the MedDRA release package. The MedDRA release package is available in the download section of the MedDRA.org website. So not everybody has access to that download section. In case you're always getting it, fine. If you've never seen this, you might want to discuss with your MedDRA point of contact whether they can download and give it to you.
And I think your audio has gone out.
The SMQ, and you can also see that here you have the narrow, the broad, but then also the algorithmic scope of the term. And then you have a short summary of the SMQ, and you have active and inactive PT counts and the active PT codes in case you're using the codes for retrieval. So now we discussed the search strategy. Of course, as Christina said, you could also look at all the terms and decide whether you want to include something else or exclude something. But be aware, if you change the SMQ, you cannot call it SMQ any longer. You then need to call it something different, like a company customized MedDRA query or whatever you come up with, but you are not allowed to call it SMQ if you change anything, because SMQ is really the endorsed standardized MedDRA queries. So then back to our data sources and search possibilities. You might have access to different databases. At Grünenthal, we had access to statistics from the Vigibase, we had the line listings from EBDAS, and FAIRS just via the public dashboard, but which anybody can use. And in the Vigibase and ultravigilance database, SMQs can be used. In FAIRS, it's really hard because you need to enter every PT by hand in the public dashboard. So for this SMQ with just 3 narrow search terms is possible, but for longer SMQs, it's really hard because you're not going to be consistent if you do it by hand. So I would not recommend doing that. From these databases, we could get line listings, but just very limited case information. I will be showing that in the next slides. What is good in these databases for us was that we had statistics available in comparison to the same PT, but for all drugs in the database. And these statistics we had for Vigibase and for ultravigilance, but we did not have access to fairs like Christina and her company. Then the question is, can an algorithm be applied if you're using SMQs in these databases? And I'm not aware that it can be, because we don't have the information and we're not able to do that. And it's not provided by the databases as such. So that then the advantages of these big databases is that in Vigibase, nearly all worldwide data is collected. So especially for a very rare event, it's good to look at the cases in Vigibase. Same for EVDAS, it collects a lot of cases from different companies. So if you have a drug which has already gone generic, you can find cases from other companies in vigilance, but with the limitations discussed before. For your company database, you might be able to use the SMQ in combination with the algorithm. You can also use combinations for other things with time to onset. You might want to look at certain patient populations. So you are free to combine any data fields as you like. And the biggest advantage of the company database is that you have the all case details, including the narrative and additional information available. Depending on the database, you can download the narrative, but it's more complicated. So these are the limitations, but also the advantages of different databases. And the next slides, I'm just going to show you what the outputs look like. It's really small print, but it's not meant to show you any numbers or anything, just to show you what kind of output you can get. And this is an example of the vigilance output for the SMQ narrow search for DRESS and for the last 12 months. You always have certain possibilities to get outputs from the vigilance, which is 2 weeks, 1 month, 3 months, 6 months, and 12 months. And but even if you take the 12 month, you will have the new cases within these 12 months, but you also have the total amount of cases. And you can see here that I searched for the level 1 SMQ narrow scope of drug reaction with eosinophilia and systemic syndrome. I just got 1 PT back of the 3, which means the others have not been reported to the vigilance within the last 12 months. Then an example of an output from Vigibase using our signaling tool, which was called Empirica. And from Vigibase, I requested an output based on the ATC3, which is anti-inflammatory and anti-rheumatic products, non-steroids, plus the PT list. So I added the 3 PTs here. And you can see that most of the drugs, you see different drugs here, all ATC3 anti-inflammatory drugs. Most of them have the PT DRESS. There's just one which also reports pseudolymphoma on this screen. And then you can see here, on the one hand, the overall count of cases with the term reported. You have the EBGM, which is the statistical measure, and then the EBO5, which is the lower limit of the confidence interval, and the EB95. The EBO5 is, as Christina said, often the threshold is 2, so 3 of the drugs signal for that PT, or the others don't. And as we are already looking, as we are a step further in signal evaluation, this EBO5 is just an information, but not really decisive, because we already decided that we are going to look at this topic, and we are looking at the cases. You can drill down into the cases in Vigibase, but you get very little information. So if you drill down into the cases, you would click on this overall number. You get an overview of including age, gender, the reporting statistics, and then the region, but not more. You can then click open one of those cases, but even there you see what other MedDRA terms were coded, the drug, also maybe other drugs, but then you don't have a narrative or any other information. So for looking at cases, this is not really helpful. This is an example of the FAERS data. It's also ibuprofen and the three narrow terms of the SMQ. And you can go to a line listing of the cases, which provide you the case ID, the suspect products, reason for use, and so on. But also here you have limited information, and especially you don't have a narrative which you can refer to. So this was just showing how you could use MedDRA to retrieve cases when you have a signal, what kind of thinking you need to apply to come to a search strategy. And this was now only focused on the safety databases, because we said that safety database is a good tool or good source of data for such a rare event. Now I'm going to show you another example, and that was dementia and proton pump inhibitors. Also here, the source was an external regulatory authority notification, and our internal signal validation body decided that this should be evaluated as a valid signal. Dementia, I think, is better known than the rest, but just to discuss shortly what we're talking about. Dementia is characterized by an impairment of cognition. Usually it's memory, but also other cognitive domains are at some point involved, like language, the visual, spatial, or executive functions. It usually is a decline from a previous level of function, and unfortunately it's often, or it gets severe enough to interfere quite heavily with daily function and independence. And we're expecting that dementia prevalence is rising substantially because of the global aging of the population over the next several decades. And when you're looking at this graph, you see that dementia prevalence starts at the age of 65, and then it rises dramatically until people are over 90. But what you also see here is that dementia is not just one disease. You have the Alzheimer type dementia, you have vascular type dementia, maybe some other in between types. So this means in a certain population, dementia is very, very common, and it's not uniform. So what does that mean regarding our data sources? The PV databases have a limitation because the drug administration is often very long before the event, so there might be a recall bias. The other limitation is that there are many risk factors described for dementia, so that a single patient analysis will most likely not provide you with definite evidence. So one case report will never be tipping it to the one side or to the other side. Then if you're looking at these big databases and you're doing statistics on these, the statistics is not driven by incidence or prevalence, but by reporting behavior. That is a general limitation for all safety databases, but especially important for something like dementia. And then you have a very high background incidence, and you would need to show that your drug is increasing the risk over that high background. So you need a lot of reports to be able to do that. The other thing which you could consider is epidemiological studies. Epidemiological studies have a big advantage that they can provide data on big populations. And you know that in this certain population, you know the denominator and you can calculate on that. But you need to be very careful to understand the data set and the biases included. And that is also true. And on the other hand, epidemiological studies need to be set up. They need a long time to run. They're quite expensive. So before starting an epidemiological study, you would first look into different data sources to decide whether it makes sense to start a study like that. So what you would look at is literature, and there maybe also focus on epidemiological studies. But also here, have in mind all the biases. Have they been addressed? Have relevant risk factors? Can they be assessed, for example, in claims databases? Often this is not even added to the database. So really important to read the literature with enough background knowledge and enough questions to say, yes, it answers my question or not. And then, of course, meta-analyses might help. Interventional clinical trials, they are most likely too short in duration and the population is small. So as this is something which has a very high background anyways, you would need to have huge studies to show a difference between the two populations, and you would have to have long-term studies. And although you might have randomized trials, there are so many different risk factors that these might not be controlled in the end anyways. So when you're looking at the demographics, you might find imbalances between the two groups. For non-clinical or preclinical data, there is a wealth of data which are more focused on describing the mode of action, but that is not well described for dementia. So also from the preclinical, it might be hard to evaluate this topic. Yeah. But from this slide, although I'm saying that everything has a lot of limitations, we are still going to focus on the PV data because that is the MedDRA coded data. Literature, you would need to discuss with your literature retrieval department what kind of search strategy you need for literature, because that is not MedDRA coded data, but there are certain thesaurus which can be used for building those searches. For ICTs, these are MedDRA coded, but as we don't expect to see much in ICTs, I will keep that on the side. And of course, the search strategy which you use on your PV data can also be applied to your interventional clinical trial data. So same as before, I would put this into the MedDRA web-based browser and would see that there is an SMQ on dementia. And because I would need to first decide whether that SMQ is applicable, making sense, I would again open the SMQ introductory guide to see what is described in the SMQ introductory guide. And again, you find a nice definition, you see the in- and exclusion criteria, and the next step would then be to review what kind of terms actually are in that SMQ. And I like the spreadsheet quite a lot, so here again, you can see that there are a lot of dementia terms which are in the narrow scope, but then in the broad scope you have quite a lot of other terms which are very general, so that it might make sense to keep with a narrow search. But for showing you what can be done in the databases, I requested the broad scope for the dementia SMQ in relation to esomeprazole from the Vigilance for the last 12 months. And what you can see here is that not that you have lots of PTs, but that there is one field which is called SMQ narrow, and sometimes it says dementia. So these are the narrow terms. Sometimes it's not included in any narrow SMQ, or you see like for hostility that this is actually a narrow search term in the hostility aggression SMQ, but not in the dementia SMQ. And when you're looking at these PTs, you're also seeing learning disability, suspiciousness, mental impairment, hallucinations, which you would not directly associate specifically with dementia. So choosing the right scope of the SMQ is also very important, so that you don't retrieve too many cases which you're not really interested in. So this was the end of our presentation. Thanks a lot, and we're open for questions.
Okay, great. Well, there are quite a few questions. So what we'll do is I'll read the questions out, and I'll also, for the benefit of the audience, paste them in the chat so people can read the question as well. So we're going to start off with the first question, which is really a two-part question. I'm going to put it in the chat right now. This question is for an individual case safety report. When an adverse event is reported, how do we say a signal has been detected? How many individual case safety reports with the same similar adverse events are required before calling it a signal?
This is a good question. So it depends really on the quality of the report and on the topic. So as we've seen, the DRESS syndrome, it's a very rare disease. So every case which is reported might be a potential signal. For dementia, which is a very common medical illness in the overall population, one case will most likely not lead to anything. Even 100 cases might not be a signal. So it really depends on the topic, on the frequency of the event and the general background population, as well as on the quality of the report. So often if you have a very high quality publication of a case report, this is more likely to lead to a signal than a post-marketing report from a pharmacist saying that patient XY had, and then just the diagnosis without any further information. So it's really not an easy question to answer with a yes/no answer. It's depending on a lot of different things. That is why we need medical scientific judgment on signals. And you would always, before you call something a signal, you would try to look into different sources. So if I see one case which is suggestive, I would look at the statistics to see, is the statistics also showing something strange? I would maybe have a very short literature search to see what is literature indicating, or go back to the previous knowledge I have of the drug. So to say, this is the first time I hear something about liver effects. What did I see in the preclinical? So did my animals, did they have any changes in their liver parameters? If they had, and that is the first human case, there is more evidence than just that case. So you're trying to gather a little bit more of evidence before you take a decision. And then if you yourself call it a signal, usually you present that information to a broader audience, which is the signal validation body, whatever it is called in your company. And that group of people then decide whether it's already enough to take action. And also this group of people needs to consider different things, that there might be previous knowledge, that it's a disease which has a very high impact on either the patient or on public health. So you would take lots of different things into consideration when you take the decision whether to do a signal evaluation. Christina, would you like to add something to this?
Yes, the GSK process, when we find a signal, we would then spend the first 2 weeks or so looking at the immediately available data to see whether or not that signal is validated. In other words, is it worth taking it any further? And we would do very similar to what you have just described, looking at, what's the likelihood? Is there a mechanism of action? Are there other products fairly similar therapeutic area that are already labeled for that particular? Or is there any previous literature that has appeared, but we think at the time, oh yes, okay, there's no signal here, but we'll monitor it, and or it might be an item in term of interest. Then again, we would take it to a small panel who have expertise in pharmacovigilance, and we might discuss with our epidemiology colleagues as well, if necessary, depending on what the topic is, before we then say, okay, this is a validated signal, and then we go into the full signal evaluation, which will then take a month or more, depending on, priority, etc. The other thing that can also affect whether or not you call something a signal is, was there a previous regulatory query from a regulator about that topic? Because it might show you that they had something in their databases that wasn't reported to the individual company. Yes, we look at the vigilance data, we look at the FDA, and Anne's company can look at WHO, but there are other regulators who also have got databases. The Australians, New Zealand, sometimes we get queries from New Zealand and Australia. Our local operating companies may say, Thailand's asking about such and such, or Japan is asking for something. So all those factors are taken into account. It's not just one factor, it's multiple factors before you decide what to do next. And if you don't do anything, again, that information that you decided not to do anything at that time will sit there. If the signal's not validated, it will be recorded. You will know what the data was at the time when you said this is not a validated signal, so that the next time that signal appears, you know how much data there has been accumulated since the last time you looked at it in detail. And obviously, if it's rapidly rising in numbers, then you might say, oh, well, okay, we'll have a look. But if the quality of the cases is poor, you still can't determine causality, because a signal is only a suspicion there's something going on. It doesn't necessarily mean that that is related to your product. So it goes through from identification of a signal, validation of a signal, full evaluation, and even at the end of full evaluation, you might decide that, oh, this is actually secondary to a condition that is already expected, already labeled, and this new signal is actually not adding any further information. For instance, if you have a multi-organ hypersensitivity syndrome and you get a report of renal problems, is that actually part of the overall hypersensitivity multi-organ? It may already be there. If your case evaluation then finds, oh, the majority of them are actually secondary to a labeled event, then you don't take it any further forward. So that it's not a straightforward answer, I'm afraid. Thank you.
Okay, Brian, do we have any more questions?
Yes, we certainly do. So I just put the next question in the chat for everyone to read. This question is for FAIRS and Vigibase. These are data repositories of past cases. So in that context, is it still a signal detection or hunting for missed signals? My confusion is arising from the fact that a signal would have been detected when the data was coming in. Now that it's become part of a database like FAIRS, at this point, are we still trying to detect signals or just trying to see if we missed a signal?
Okay, I understand that question in a way that you're asking if you are sending your cases into FAIRS and you've reviewed those, that you would not make them a signal from FAIRS. So it really depends on where you do your signal detection. Of course, like Christina showed to you, that statistical signal detection, it often stays up. So for the example of tinnitus, since 2018, Christina has been seeing high statistical scores for that topic. And that is something which we also used to see, that once you have a statistical signal in a database, it usually stays a signal. It's not going down again. So you would do your signal, you would look at that data and decide whether you have already so much previous knowledge that it might be even a labeled event. And in case of a labeled event, you would need to decide, is there something new, a new aspect to this, which I expect, which I would like to review? Or you could just say, oh, okay, this is something which I now see it in that database as well. So this is not of concern to me because it's a now known, which we discussed in the beginning. It could also be that you see it, but you have some new cases reporting slightly different information, so that you consider reopening something or looking at it further time. Could also be that you looked at the statistical signal at some point, you even did your evaluation, but it was all confounded by other things, by other drugs, by other risk factors, so that you decided, oh, it's still not, there's not enough evidence to say that my drug is really causing it. I've just seen it in combination with this or that. But then you might receive new information saying that, oh, there are cases where it's just your drug reported without the other drugs, without those risk factors. So you might need to look again. So it really depends on the situation and on the previous knowledge, whether you would consider something you see in FAIRS again as a signal. Most likely not just by seeing a continuous high statistical signal, but then having some more information which makes you suspicious that it might have changed a little bit. There might be other information which I've not considered, so that you look at it again. Or you could have the situation where you first looked at a signal surpassing this threshold, a statistical signal. You decided that I'm not so sure that there is enough information yet, but then you can also already decide that after a certain period of time you would like to look again to see how did the information change. Is it still the same information reported or not? So if you're a little bit unsure, you might even decide to keep that under closer monitoring and to look at your case, your incoming cases, look at how that signal changes, look at the literature for that topic with a particular interest, and then to see whether you find something which makes you reopen. So to implement it into your continuous signal detection as a special topic of interest, and then once you find that the information changes, that there is enough new information, then to look at it in full again.
One other thing you might want to look at, if your drug has an indication for two different patient populations, you might want to examine your signal by indication. In other words, split the data and review each indication separately and see whether or not it is actually coming from one patient population but not in the other. Because if it's entirely coming from one patient population, you would think, what are the risk factors that have made that particular patient population report this problem, but same drugs being taken by another patient population, a separate indication, and it's not appearing. The other thing is there a difference in the dose between those two patient populations? Is that what's causing it? Or is it an underlying tendency for one particular patient indication, a patient population, to develop that kind of problem.
Okay, we'll move on to the next question. So the next question is, I'll put this in the chat here. I'll send it to both internal and external parties. I saw your message, Anna. This is, first question is, approximately how much time should each step in the signal detection process take for a company?
There are timelines, and it's again hard to give just one answer, because there are these things which Christina touched upon shortly that would lead, just one case could lead to really measures being implemented directly. So these are called emergent safety actions, and they are often, the companies have procedures which just allow for 2 days, I think, for 48 hours or 72 maybe, but really very short timelines if the public health impact is very high. For other things, you need to consider that you have, depending on the region where you're operating, but your signal will potentially lead into label variations. And once you've made it a signal, the evaluation and the label variation all need to be done in a certain time frame. So you need to consider several things on how much time you can spend in which step. And if you take more time in the beginning of the signal detection or signal evaluation, then you might have less time in the end. So that is things which you need to consider, but it's also depending on the medical importance of the topic. So if it has a high public health impact, has a high patient impact, then you need to do it quicker than if it's of lower importance.
And in addition to the actual signal evaluation and getting a decision, you also need to consider, when you put it out for labeling, what is the patient impact of the speed of labeling. So for instance, if it's a contraindication, that label has to go out very quickly to your operating companies and communicate to the regulators very quickly. If it is an ADR that has relatively minimal, okay, say alopecia, which is loss of hair, it's not good for the person's appearance, I'll give that, but you know, compared to liver failure, what is loss of hair? So one thing might mean you have to contraindicate, do not use in that patient population because it causes liver failure, whereas compared to that, you give this to particular people and very rarely you will get some loss of hair. So the time at which you communicate that can also vary, and within some companies there are guidelines on how quickly the communication takes place.
Okay, we'll move on to the next question, which is, do you take data from clinical trial and post-marketing data as two separate cohorts for signal detection?
Well, yes and no. We had some of the SIEs from clinical trial, we had them in our safety database as well, but we did not have merged databases with the clinical database. So we could always, retrieving data from our safety database, we would have very limited clinical data in there in the sense of SIE reports. But for looking at the complete clinical trial data, we needed to go through our statistics department to look at one clinical trial, or if we were lucky, we had a pool of clinical trials which we could look at the same time. Usually, if you're looking into the clinical trial data, it's not as fast as looking at the pharmacovigilance databases. Pharmacovigilance, we had our own personnel retrieving the cases with quite easy case retrieval logic. If you're looking at clinical trial data, especially if you still need to do some pooling of the data, you need to consider a lot more time because first of all, the data fields in different clinical trials need to be put into one database or one depository. They need to be made at the same field in one clinical trial is compared to the same field of another clinical trial. So it needs a lot more data transformation if that was not done previously. And also the outputs are programmed in a different way. So usually it takes a longer time to get the clinical trial data. Sometimes if you're lucky, you already have a pooled analysis, which is often done after phase 2 and once again after phase 3. When you're looking at the complete set of data until that point in time, to take further decisions for the clinical development or after phase 3 for the marketing authorization procedures. Then you often have a good pool of data, which is more easily accessible, or you already have outputs, which you can use also for safety questions later on, for example.
It's very much the same situation as Anne described. If you've got a product that's already on the market or recently on the market, then in addition to the post-marketing reports that you will start getting, you will have a very nice integrated safety data set that was used for the submission to get the marketing authorization. So that is extremely helpful. You can't merge the two sets of data, post-marketing and pre-marketing, for signal detection as such. It is a manual judgment process, purely because they are very, very different. Post-marketing data does not have a placebo or a comparator group. You just have poorly documented, very often, spontaneous reporting. So a pharmacist says, patient X came to see me. He said he had spots. What kind of spots? You don't know. What type of rash? You don't know. Whereas in clinical trials, the investigator will have documented the type of rash and the severity of the rash. So it is very, very different data, and it can't be merged without a lot of thought.
Okay. Okay, we'll move on to the next question, which is, in many PSURs, it has been observed that data in post-marketing is spontaneous, or the spontaneous reported data is low in number. Isn't underreporting a concern?
Yes, there are lots of concerns with the post-marketing data. Underreporting is one concern. And then also, if there is, it's called stimulated reporting, if there is media coverage on a certain adverse reaction for a certain drug, then it's reported a lot more than without that media reaction. Especially in the US, there are sometimes litigation cases for certain drugs and a certain event. And then pharmacovigilance departments are overflooded with cases of very limited quality for that event. And so there are lots of drawbacks with spontaneous data. But of course, this is a data source which we are collecting, which gives us information, and which is very helpful for those rare events. Dress is one of those events which is very hard to evaluate in any other data source. Same as other severe skin reactions. very severe liver failures are often first reported in PV data. But it's a very critical data source for common medical conditions. So if there is a high background, it's really all these drawbacks are very important, that you don't have a comparator, that you have underreporting, that you can have stimulative reporting. So there are a lot of drawbacks with the PV data, with the spontaneous data.
Yes, entirely in agreement. There are serious drawbacks. Another advantage of PV data, even though it is underreported, is that during clinical trials, you have a very controlled patient population. You have inclusion and exclusion criteria, so it's clean data. You don't know what's going to happen after it's been marketed and people are taking all sorts of concurrent drugs that were not permitted during your clinical trials, for example. So you get unexpected reports of potential drug interactions. There is no CYP enzyme that is, in common between the two products. You might have studied it in the PK studies and the PD studies during ClinPharm, but you do get unexpected combinations in post-marketing. The other thing is that people take drugs off-label. No matter what you tell them, they will take drugs off-label, and it's prescribed off-label as well in some countries quite a lot. So you get patients who have received that drug who were not the target population during your clinical trials. So PV data can be very different, but it can also be extremely useful.
All right, next question is, how do you deal with active surveillance and passive surveillance data for signal detection?
Christina, would you like to answer that? Because we did not have much active surveillance.
Active surveillance is where you have identified an issue and you are targeting that particular event. Usually, in regulatory terms, they would be called enhanced pharmacovigilance plan. So you have a risk management plan for your product. You will have an enhanced one, a special measure. In other words, you're doing over and above the normal passive surveillance. So, for instance, if your product is, say, an anti-epileptic, and you have you're monitoring sudden death, which does occur in the epilepsy population, unfortunately. It's very difficult to get information from a reporter when they said somebody's died suddenly. They've not had ECGs done beforehand. You don't often know what they've taken. There may not be a post-mortem. So there are some times when you just have a dead end, you can't follow up. On the other hand, if it's something, say, serious adverse skin reactions, then you can, when you get a report like that, go and actively send a detailed follow-up questionnaire and collect that data from the reporter. And if the reporter's consumer, you can ask them for permission to contact their doctor and send that form on to collect actively more information. There are also different programs for risk management that you agree with your licensing regulator for these additional management plans. And that might mean further education, for instance, roll out, don't increase the dose above this particular level and at this particular titration rate, and then monitor the number of adverse reactions you get. that you're trying to prevent to see whether or not your education plan works. So there are all sorts of ways of doing active surveillance, but usually active surveillance is something that you would agree with the regulator who has licensed that drug, and it will be described in your PSURs as well and in your risk management plans.
Okay, are we ready for the next question?
Okay, go ahead.
All right, great. So the next question is, for relatively small companies with limited products and no access to your due vigilance, any suggestions on how to conduct quantitative detection?
Well, the problem is always that if you want to do a quantitative signal detection, you need to have a good background, and you need to have a big safety database which you can use as a background. So if you don't have access to any big database, maybe you're not. There is no good way for you to use quantitative signal detection. Christina, do you have any good ideas on how to do it without data?
Literature is a possible source, and obviously get access to EudraVigilance and WHO.
And I think if you're operating in Europe, you can have access to EudraVigilance for your products which are authorized in Europe. I think there is no limitation. I don't think that we paid for it. Vigibase might be a little bit harder because you need to pay fees, but it always depends on whether you can get that access or not and where you are located. So EudraVigilance should be a possibility if your products are registered in Europe.
Okay, let me move on to the next question. This one was, the next few questions are about the threshold. This one here is specifically addressed to Christina, which is, I lost it here. Who defines the threshold? Is it the regulatory body or the pharma company by themselves?
There are known thresholds which have been published by signal detection experts, of which I am not one of them, as I certainly don't work on the statistical, the numerical side of things. I'm a physician by training. But signal detection methodology is in the literature, and the FDA have decided to use the Bayesian theory, which is using the EB05, and that is the confidence intervals of EB05 being 5% and AB95 being 95% confidence intervals. And that threshold has been set at 2. The EudraVigilance database uses a different statistical method. They use the ROR, which is a reporting odds ratio, and that has been set at 1. Now, why they were set at 2 and at 1, I'm sorry, you're going to have to take it to a statistical safety detection expert, and it is available in the literature.
Unfortunately, I'm not that expert either. But I think you, as a company, you're not forced for any of these thresholds, but of course you should have a good argumentation why you use a certain threshold. And EB05 of 2 was shown to have a good balance between false positive and false negatives. So that is a threshold which is quite widely accepted. But for the basic, science behind that, I would also need to refer you to literature or experts. Brian, do we have any more questions?
Sorry, I dropped out there. Are we ready for the next question? Yes, sorry, I dropped out for just a second. Yeah, the next two are, again, related to the threshold, which the first one here is, can you provide more information on how you determine the threshold?
This is not completely clear to me. So as a company, you would need to decide for your in your signal detection process which threshold you want to look at, and that is something you would need to look at the literature. If you are using a certain tool to look at the data, then the technicians will put the defined threshold into that tool. And as Christina said, for certain databases, there is an established threshold, like for the FDA, it's the EB05 of 2. For ultravigilance, there is a certain threshold. So this is already built into the system by the during the technical setup. So when you're reviewing the data, you usually have, on the one hand, you have that threshold in mind, but some of these tools also help you and show you the data with this line where the threshold is. I don't know whether I understood the question completely. Maybe Christina, your thoughts on this?
As far as I know, the tool that we use is off the shelf, and it's been programmed by the software companies that sell you the tool. And I believe you can customize the tool as well. So you can go to them and say that your statistical expert wants to use this particular, use the ROR like the ultravigilance instead of what the FDA uses, which is EB05. So you can choose what statistical method you want built into your tool. And once it's built in, the user only gets the figure for each PT at the time, at whatever intervals you want the data loaded. So you need to have an interface between your pharmacovigilance person and the person who's controlling the tool. And very often that software tool is, there are companies that will do that off the shelf, but you can also customize it to individual company requests.
Okay, I think we're going to have to start wrapping up there. We have got just a couple more slides, but I just want to say thank you very much to Anne and Christina for a really fantastic presentation, really clear and simple for me, for your explanations and all the great examples to put things into context. I'm just going to talk very briefly about what would you like next. What we've had a great subject today. But what else would you like to see in one of these webinars and future meetings? Please get in touch. We really would like to hear from you on all the topics that you feel would be useful. And just a reminder that this presentation is in the handout section of the webinar panel on the right-hand side of your screen. And we'll also be posting on the MSSO website, as well as the recording of this session on the YouTube channel. And then finally, the last slide is just a quick mention of our upcoming user group meeting, which will be an in-person meeting in Amsterdam, and that will be happening in April after Easter next year. More details to come, and there will no doubt be some mail shots and information on the website. So with our last minute, I just want to say thank you very much to the presenters and the panelists, and thanks for the organisers. The MSSO have been fantastic yet again, and thank you very much for all the questions. Sorry we didn't get around to answering everyone, but thank you very much for submitting your questions. Really appreciate everyone's attendance today.